Okay, this is the second test we had, and here we go. All right, so this is the tour of the cell. Remember, cells are very, very, very small. You need microscopy to see them. Different kinds of microscopy exist. Cells are very small because they must keep their ratio of surface area to volume very, very big, right? We need big surface area to volume in order to get things out and into the cell. So make sure that you understand that concept. Larger cells, less surface area to volume ratio. So we want small ones. Cell theory, all cells are made of one or more cells. Cell basic unit of life, cells arise from pre-existing cells. It's very important that cell structure is correlated to cell function, as you guys saw in your cell projects. The basic features of all cells: phospholipid bilayer, cytosol, some sort of genetic material, and ribosomes, which help make proteins from RNA. So remember, there's always a flow of information: DNA to RNA to proteins. The plasma membrane is dictated by a fluid mosaic model where we have the phospholipid bilayer formed by those nonpolar tails coming together with lots of proteins stuck in there as the little mosaics. Prokaryotic versus eukaryotic, you've got to know the difference between these. Much smaller, no nucleus, no membrane-bound organelles. Eukaryotic cells, larger nucleus, lots of organelles. They all, again, if you look at just the eukaryotic cells versus there's animal and plant cells. Animal cells have lysosomes and centrosomes, centrioles, flagella, whereas plant cells have chloroplasts, central vacuoles, and cell walls, and plasmodesmata, the little pores where things can go through the cell walls. Plasma membrane is selectively permeable barrier, right? That's something you should say if there's a free response about that. Selectively permeable barrier. The endomembrane system is also really important for different membranes in the cell. That's how we get phospholipid bilayer. That's how we get a lot of the cell. So the endomembrane system, I'm not going to draw it, but it has the nuclear envelope going to the ER, going to the Golgi, going to out of the cell. And then we have lysosomes, vacuoles, and vesicles in there as well. Eukaryotic information, genetic information, again, always starts from DNA, which is in the nucleus or the nucleoid region, going to the RNA, which happens through transcription. RNA then goes to proteins through translation. DNA is organized into chromosomes. Prokaryotic cells, a single circular chromosome, lots of linear ones for eukaryotic cells. Eukaryotic nucleus is pretty complicated. We have two lipid bilayers, an inner and an outer membrane. The outer membrane is continuous with the ER, and there are nuclear pores to get things in and out. One of the most important things to get out is mRNA. Ribosomes are the protein factories, these little Hawaiian buns. They are made of RNA and protein. They exist freely in the cytosol and bound to the ER. The endoplasmic reticulum, or the ER, is part of the endomembrane system. Huge amount of membrane. It comes in two flavors: smooth for lipid synthesis and rough for making proteins. Sorry, there's a sliding glass door opening and closing. The Golgi apparatus is for shipping and receiving, puts little tags on things, right? Little sugar tags. So it's involved in adding sugar tags and telling things where they need to go. The Golgi has a cis and a trans face, and they're folded into cisternae. Baby, you gotta stop with the door. Lysosomes are digestive compartments. This is where digestive enzymes, it has a low pH, it's like the cell's stomach. Vacuoles are food vacuoles from phagocytosis. Contractile vacuoles in many protists to pump water, and central vacuoles in plants. The energy converters are the mitochondria and the chloroplast. Mitochondria are responsible for cellular respiration. All eukaryotic cells have mitochondria. Chloroplast, plants only, photosynthesis. Got to know the structures of these two organisms. You need to understand the endosymbiont theory about how prokaryotic cell was phagocytosed by a eukaryotic cell to become these organelles, to evolve into these organelles. A mitochondria was not engulfed, all right? It was a prokaryotic cell, which then evolved into a mitochondria within the cell. Peroxisomes, boring. Breakdown of long fatty acids produce hydrogen peroxide. Cytoskeleton, one of my favorite parts, made of 3 different types. You have microtubules, big hollow structures, actin, thin ropes, and intermediate filaments, which are intermediate size. They help maintain the shape, cell motility, mechanical support. Centrosomes and centrioles are at the heart of a centrosome. The centrioles are at the heart of the centrosome. They help maintain normal cells. They form the mitotic spindle, and they are very important for making cilia and flagella, where they act as basal bodies. Cilia and flagella are important for moving. There's also the extracellular matrix. We have cell walls. We have different junctions holding cells together. So there's lots and lots of parts of the cell. All right. Then we are going on to chapter 7. Just rolling right along. Where we talk about membrane structure and function. So the fluid mosaic model, remember, we kind of talked about this last time, or in the last slide. Selectively permeable barrier. You need to know what can come in, what can diffuse right through, meaning small molecules, uncharged gases, whereas other things, larger or polar, they need a different way to get through, either facilitated or active transport, right? So diffusion always goes from a concentration of high to a concentration, from a high concentration to a low concentration, down their concentration gradient. We see this in the lungs. We see this in a lot of different examples. Remember these kind of U-tube examples, and osmosis is specifically diffusion of water across the semi-permeable membrane, right? So here, for example, only water can get through. So if we have a hypertonic solution, meaning there's more solutes, water is going to rush over. The salt, putting salt on a slug, that causes all the water to go out towards that salty environment, down its concentration gradient, and it can dehydrate a slug. Remember that we have hypo, and isotonic solutions, right? Hypotonic, water will rush in. Isotonic, same. Hypertonic, water is going to rush out. Remember, whenever I see if something hypertonic, I put a bunch of solute there, and we know the water is going to go towards that solute. Membrane transport can either be passive, meaning down its concentration gradient, takes no energy, or active, against its concentration gradient, needing ATP. Passive diffusion is diffusion directly through the membrane, like small gases, steroids, etc. And facilitated, it's going through a membrane protein. Active transport, you need ATP. It's going against the concentration gradient, or you can also have bulk transport. So facilitated, going through a protein. There's different ways that you can have facilitated diffusion. It can either be a protein channel or a carrier protein. If you involve the hydrolysis of ATP, meaning you are breaking one of these bonds in ATP to make ADP, you are getting a lot of energy being released. That is an we're not there yet. You get a release of energy, right? So in active transport, you are moving things against their concentration gradient. The best example of that is a sodium-potassium pump, where you're moving sodium out against its concentration gradient, and you are moving potassium into a cell against its concentration gradient. Takes a lot of ATP to do that. You also have proton pumps, which we see in the mitochondria and in the thylakoid membranes, right? In the mitochondria, you are pumping hydrogens into that intermembrane space. In the thylakoids, you are pumping hydrogens into the thylakoid membranes. These create electrochemical chemical gradients, which will be very important for making ATP later. There's also cotransporters, where here we're using ATP to create a hydrogen gradient. We are then using the energy stored in that electrochemical gradient to pull sucrose across. Then we have active transport, the ability to exocytose or endocytose, meaning get rid of things or take things in. There are 3 forms of endocytosis that we need to know about. Phagocytosis, just engulfing big particles. Penocytosis, drinking liquids. Or receptor-mediated endocytosis, meaning we have a ligand and a receptor necessary to form that to induce endocytosis, right? Again, ligand binding receptor causes that formation to change. All right, then we have, where's my enzymes? We'll stop this one here.